Um, Good evening, everyone. Good evening from Boston. My name is Nicole Dusang, and this evening's BCI Thursdays event is part of the BCI Society's Next Generation series. As the name implies, this workshop series was created for the next generation of researchers and investigators. The aim is to provide technical background on cutting-edge topics in BCI research, and most importantly, to answer questions that trainees might have. Tonight's workshop will be presented by Jason Palmer of Osaka University, where it is actually Thursday. Professor Palmer is an expert in statistical signal processing and machine learning methods and techniques. And this evening, he will discuss EEG data analysis for BCI applications.
Okay, thanks a lot, Nicole.
Yeah, no problem.
So it says host disabled participant screens sharing. Uh-oh. I think I got it.
Are you having problems sharing your screen? They said it should work now. I have just one more announcement for all the attendees. Please use the Q&A for your questions. That's going to be monitored for the Q&A portion after the presentation.
Okay, can you see? Excuse me.
Yes, we can.
Okay, great. So this is basically going to be a very basic introduction to EEG, particularly with application to BCI. Forgive me if you know a lot of this stuff, but I'm going to sort of try to go over most of the basics. Specifically, we're going to look at what we know that EEG is a tool for measuring brain activity. And so first we're going to look at what kind of brain activity EEG is actually measuring. And then briefly, how does EEG measure the brain activity? And then what kind of applications can EEG be used for, and how can EEG be used for those applications? So first, I hope you can't see this. Okay. What kind of brain activity does EEG measure? So we know that neurons make up the fundamental sort of electrical unit in the brain. And we know that neurons generate this large action potential, but actually it turns out that EEG is not measuring these sort of this neural firing directly, where you're getting these large action potential jumps registered by the EEG. Rather, it's measuring ion movement, ion flux in the extracellular fluid. So the ions that we're talking about specifically are sodium here, which has one valence electron, which it loses to become a positive ion, sodium, potassium, calcium, which has two valence electrons, and then chloride, which is missing an electron, which it gains to become chloride. So these are the four main ions that are sort of swimming around in the extracellular fluid. Now, if we look at the cortex, it generally structurally consists of six layers of different sort of types of neurons, granular or pyramidal neurons. And then there's sort of extracellular fluid, sort of ion bath, sort of where these neurons reside. And it's thought actually that as the neurons fire, they take up sodium. So they open sodium gates and sodium rushes in. And it's thought that, so this is an example of ions sort of moving during activity. So it's thought that when you have many neurons synchronously firing, they take up a lot of the ions in the extracellular fluid, particularly what's seen in EEG would be what's taken up into layer 1. So this figure sort of represents ions, sodium ions here, for example, in the extracellular fluid, and then being taken up by the neurons in layer 1, and then what the electrode sees is a relative reduction in the potential. So this is basically this movement of ions that in the vicinity of the electrode that is thought to make up the EEG, the potential, the fluctuations that are measured by the EEG. So in addition to the influence of the local neurons firing, we have these long-range white matter connections, which connect, say, the frontal and the parietal regions, and also they connect subcortical regions like the thalamus to various regions like the occipital area and the parietal, the frontal area. And we have the cingulate tract. So these structures, these white matter connections influence can influence the coherence, basically the sort of synchrony of activity at long range brain areas. So at the level that we look at where we're measuring EEG, we basically are looking at the level of this sort of large-scale functional organization of the brain, where we have the primary motor cortex and then the somatosensory cortex involved in movement and touch, and then we have the frontal area involved with executive function, planning. We have the auditory area. We have the parietal area, which is very relevant, especially for BCI, in that this is where sensory information is integrated and associated. So it's often where if you detect a target in the sense of, say, the visual sensory area modality, you get a potential in the parietal area, representing the sort of detection of or the pattern matching of the target with the the pattern. So moving on, so that's sort of what EEG is measuring. Moving on to how EEG is measuring it. The classical way for most EEG setups or caps that you would buy is to use these passive electrodes, which are normally made of silver and silver chloride. So basically the way that these electrodes work is they require, in addition, an electrolyte gel, which will contain chloride ions as well. So this gel interfaces with the tissue, and in the case of EEG, with the skin of the scalp, and then also with the electrode. And so, as we said, we have fluctuations in the ion potential near the scalp, and this creates fluctuations in the electrolyte gel. So what happens is you get a oxidation of the silver in the electrode, which a silver ion entering the gel, and then an electron leaving on the electrode wire. So that creates a flow of electrons, say, down the wire, and then as the tissue becomes negative, so you get so as the tissue is negative, it draws the silver ion out of the electrodes. As the tissue is positive, it pushes the silver ion back onto the electrode, and that causes the fluctuation in the current of the wire. So that's the sort of mechanical, chemical way that the brain ion fluctuation is transferred into the electrical current in the EEG wires. So there are actually newer types of electrodes. In particular, there were active electrodes that, since the EEG signal is so weak, and in particular we have on the wires, we have power line noise, either 60 hertz or 50 hertz. And so active electrodes attempt to drive a current through the electrode and the body to cancel out this power line noise and greatly increase the signal to noise ratio of the EEG. So one of the most popular companies and products are the biosemi electrodes, which are active electrodes, and you can find their publications on the particular circuits they use at this website. Also, there are dry electrodes now, so a lot of companies trying to avoid using the gel, which is necessary for the classical sort of passive electrodes. So again, rather than just being sort of metal electrodes, they contain circuitry and amplifiers and things on the circuit, on the electrode itself, and so they become rather complicated. But one company that seems to make a decent dry electrode is the Cognionics company, and they have links to publications and products at this website here. So typically with EEG, we have a set of these electrodes at standard locations. So the C stands for center, F is frontal, P is parietal, O is occipital. So generally there'll be a reference given in an electrode cap, which is can be any number of channels, but up to basically 256, and then the channels are basically interpolated in this classical system. So this is called the 1020 system montage because of the percentages of the distance of these sort of key electrodes. So you first find what's called the nasion between the eyes, and then the inion, which is the base of the skull, and then you place CZ, C or CZ, exactly halfway between the nasion and the inion, and then you've got these 20% distances, and then the 10%. So that's the 1020. So another thing that that comes up with respect to the sort of montage is that you can get in EEG a pattern on the electrode, say, and then what you do is try to solve an inverse problem of a source being inside the brain, passing through these various layers. In particular, the classical model uses skin, skull, cerebrospinal fluid, and brain tissue model. So that the CSF is in the subarachnoid space here. And then there are more complicated models that are not, that have either a specific average head shape or a head shape based on actually an MRI of the particular patient subject. And then you can basically try to find the dipole, sort of little dipole location and orientation that minimizes the variance with a potential pattern of EEG. So this is an example of EEG Lab, where which is an open source software that based on MATLAB, which localizes this pattern to this particular dipole with a residual variance of 2.75%. So the EEG will be, the EEG will be after so being recorded by the electrodes, it will be amplified 1,000 or 100,000 times, and then it's saved at, it's then sampled at a particular sampling rate, typically 1,000 hertz, because EEG doesn't measure above 1,000 hertz, it really can be limited to almost 100 hertz, but 1,000, so you get like 1,000 or 10,000 as the sort of basic sampling rate of an EEG system. So then the EEG is filtered with an anti-aliasing filter before sampling to prevent aliasing. And as I said, the maximum EEG frequency is, is typically less than 100 hertz. So it can be common to reduce it to 256 hertz, reduce the sampling rate so you reduce the size by 1/4. But basically, you can always do that as long as you reduce to a sampling rate that's twice the maximum frequency that you're interested in. That's the Nyquist rule. So turning to BCI, we can use EEG as a measure of brain activity for most applications in BCI. So, for example, spelling devices where a sort of cursor will move across potential choices in an alphabet, and you respond with a switch or a click, or you can have multi-dimensional cursor control for sort of more free movement in a computer interface. So that's more of that would be a sort of classification problem. And then it's also possible to control devices, prosthetic devices, or assistive sort of prosthetics using EEG. So some of the advantages of EEG are that it's has very high temporal resolution, as opposed to, for example, fMRI. It has a millisecond temporal resolution, so it's basically recording things at the timing that the brain is making its decisions, and is being modified. And then it's a relatively sort of low cost and portable. Some of the disadvantages, though, are its sort of low spatial resolution. EEG in particular, with respect to this, due to the skull, has a low sensitivity to high frequency activity. So it's so it has mostly sort of lower frequency activity, and it's sort of smoothed by the traveling through all the layers and the skull. So basically what we want to do in BCI can be formulated generally as either a classification or a regression problem. So in the case of the EEG, we want to map this multi-channel EEG activity at the current time, which will generally include some past of, say, 500 milliseconds or 1 second, So like a window, so it's a sort of short matrix of activity, and we want to regress that or onto some signal, say a robot arm trajectory, or I classify, say, a particular movement uh, type or some other type of classification. Um, so basic, excuse me. Basically, we want to use this data here as the input to a classification or regression problem. So our problem then is to avoid overfitting. So basically, using EEG for BCI comes down to determining what kind of features we can use, or what kinds of ways we can transform this large dimensional data into a lower dimensional feature vector that can then be used in with any number of the classification regression, say SVM or deep learning type methods. So with EEG, we have basically these three types of features. Let's say amplitude features, where we're just looking at the potential at, say, a particular electrode or subset of electrodes. Or we look at, say, a component source signal amplitude that's formed by either a beamforming spatial filter or an ICA weights filter, which I'll just discuss in a minute. So we can use these amplitude features as the input to a sort of thresholding system that, for example, the speller will use the P300 as input to the sort of click system. So I'll discuss that in a minute. Also, we have spectral features, which are sort of power in various frequency bands. So again, this will vary over different channels, and then we can also use different. We can also use these beamforming or ICA signals, and then various connectivity measures. So I'll talk about these briefly. So the ERP is an average over a number of trials, all locked to a particular stimulus, and usually with a baseline period average removed. So this, as I was mentioning, is the P300, which has a peak around 300 milliseconds after the target, as opposed to a non-target stimulus. And so we could use, we could detect a target, say, using a threshold of 0.4, say. So these are usually measured in, as I mentioned briefly before, the parietal electrode, which is the sensory associative area. So it is sort of comparing the sensory data to a target pattern, and then responding apparently if it matches and with increased neural activity, which creates this this peak. So for BCI though, we don't use actually the ERP, obviously, which is an average. We're going to be using just the activity, say the the PZ electrode itself, but so which won't always be a nice peak, like when there's a target present. Sometimes it'll be interfered with and won't necessarily look like this, but we generally we like to look at ERPs to try to detect when there's some kind of significant response to a target signal or a target stimulus, and possibly different types of waveforms. So if there is a significant response, there will be a sort of some kind of waveform here that has a significant amplitude, because this is an average. If there isn't a sort of a regular waveform that occurs to this stimulus, then this will be 0. Sorry, did I forget what I said? But if there is one, then this will average to something non-zero. So you can look at the ERPs to the different channels or different components and sort of limit the channels or the components of interest to those that show significant amplitude response to the target stimulus of interest. So with EEG, you usually hear about the different frequency bands from delta, the very low frequency, 1 to 4, the theta 4 to 8 hertz, the alpha, which is typically in the occipital area, reflecting the absence of visual activity, either when the eyes are closed or you're looking at a blank screen or anticipating visual input. Beta activity is usually seen with actual either neural activity, or it can also, beta oscillations can reflect motor inhibition activity. Then gamma is associated often with local neural firing as well. So we typically see activity in these frequency bands. They're not arbitrary. They're created because activities, for example, the theta activity will be associated with actually concentration or memory access, and the alpha will be, as I said, associated with this sort of visual idling process. And so when we're interested in these different things, we can limit our consideration to different frequency bands, or we can create average power in all of these frequency bands, say that to then have say 6 degrees of freedom, as opposed to the full, say if we did 256 point FFT, we have 256, 128 degrees of freedom. So in addition to the ERP, we have the spectral features, the spectra, the spectrogram, and the ERSP. So the spectral density is basically the plot of this distribution of power as a function of frequency And this is taken over some period of time. So usually it's sort of relatively long period of time. So for BCI applications, we're interested in short time sort of changes in activity. So we're really interested in how this spectrum is changing. So generally we view the spectra as a spectrogram, which is a time frequency plot. And so this is an example of the time frequency plot, and it's actually an ERSP. So the ERSP removes the baseline period. So the ERSP stands for event-related spectral perturbation. So that means it's how the spectrum is being perturbed by the event. So an event, in this case, as usual, it takes place at time 0. So in this case, this is yeah. So this is the stimulus occurring at when the plus, or when the X comes on in the video, and then the task is to move the robot arm here, either flexing or extending the hand. So that X occurs at time 0, and what we see is basically this beta activity and alpha activity being suppressed. So again, this is a perturbation, so we're subtracting out the baseline, so it's relative to baseline. So this is all, the blue is, when the power is decreased relative to baseline, and the red is when the power is increased relative to baseline. So this shows a decrease in power in the theta, alpha, beta range, and then an increase in power in the gamma. So another technique that's often used with EEG these days is ICA. So this was originally a sort of audio source separation technique for separating multiple speakers using multiple microphones. And Scott McKay, who I worked for and with at UCSD, with Tony Bell and Terry Sinofsky, applied this technique to brain EEG data and found that it gave these nice sources. So it's thought, again, that these sources are these local patches of synchrony, generally oriented perpendicular to the cortex. And we have multiple sources in the brain coming from the different regions, possibly coherent with one another. So the ICA model is basically a linear generative model of EEG, where the EEG data here is this matrix of channels by time is set equal to a set of what are called component maps, or sort of these average potential patterns across the electrodes times these driving sources, which are represent the electrical activity that together with this this sort of average potential map that it's driving is visible in the EEG electrodes. So the model basically looks like this. We have this matrix here, which we're going to try to decompose into a square matrix of maps times the set of driving independent components. So this is an example where we have the raw EEG at the top, which is a mixture of multiple sort of signals. And then after running ICA to decompose this, we separate out the ocular eye blink activity, and this is theta activity here. This is alpha. These are alpha bursts associated with the screen coming on and off. This also theta activity alpha. This is actually the heartbeat signal, which is separated out, and then this is scalp muscle activity. So this is actually not brain activity, but it shows up in the EEG because it's electrical activity, but it's actually muscle activity in the scalp. So basically, the ICA works in this sort of linear analysis synthesis method where we have an analysis matrix W, which is the inverse of that model mixing matrix I mentioned, which is applied to the scalp data to create the source activations. And then the data itself is modeled as those source activations times the mixing matrix of independent component maps. So finally, with connectivity, we have a number of measures that are often they're used most often. So the first one is the phase. Sorry, this should be. The phase coherence. So we can estimate the instantaneous phase. So I'm just going to go over this briefly, just to give you an idea of what is used. So we can estimate the instantaneous phase which is sort of the position of a sine wave along the time axis. So we're interested in the relative phase. So we're interested in the phase difference between one sine wave and, let's say, at one location and the sine wave at another location. So that will generally be some value between 0 and 2 pi. So it's represented as an angle in the complex plane. So one of the most commonly used measures of connectivity is the phase coherence, which is essentially you take the windows, a bunch of windows of the two signals, and you get the phase angle difference. And essentially, you can imagine them all being plotted on this circle. So this is a complex exponential. And then the phase coherence is the average. So it's going to be some value here, and it's actually the magnitude of that is the phase coherence, and the angle is the phase lag. But if there are, so in this case, there are this phase difference is you can see it's concentrated in this area here. if it weren't concentrated, it would be distributed around the circle, and the average would be close to 0, and the magnitude of the coherence would be 0. So it's always something between 0 and 1. Now, another measure is introduced by STEM. So you have to be careful, because sometimes this is called the phase locking index. So these acronyms aren't always used consistently, but the phase lag index defined by STEM is actually the sign of the difference. So, or the average of the sign of the difference of the. So in this case, there's a definite phase lag here. So the sign in this case, it's positive if it's on this side, and negative if it's on this side. And if we average these, we get a positive, relatively positive value here. In this case, representing the connectivity here. Whereas if they were distributed randomly, the average would be thought to be close to 0. Finally, there's these more complicated connectivity measures based on multi-variate autoregressive or vector autoregressive modeling, which estimates the number of or P matrix coefficients representing the connectivity of a number of multiple multi-vector signals. And generally, you can see the formulas here in the frequency domain. So you formulate these matrices, and what's called the directed transfer function is defined in this way, where these H vectors are the vectors of this H matrix, which is the inverse of this, the D inverse transfer matrix. And then another measure more recently proposed is the partial directed coherence, which is actually defined in terms of similar components or similar parameters, but it just has this form, and it's been shown to have somewhat more interpretable coefficients of connectivity of different sources. So just to summarize, the EEG measures these fluctuations in the ion concentrations around the patches of cortex with synchronously firing neurons. So again, it's the ion fluctuation, not the neurons firing, that the EEG is measuring. But long-range coherence can result from the white matter connections, long-range white matter connections between different cortical regions and with subcortical structures like the thalamus. So generally, we use these AGCL, silver chloride electrodes, and electrolyte gel, and which exchanges the silver ion with the electrolyte gel, although there are new dry electrode technologies being advanced all the time. And the forward models can be used to solve the inverse problem of source localization for given EEG patterns. And we looked at different features that can be used in the BCI problem of either classification or regression, including their ERP, the event-related potential, the ERSP, event-related spectral perturbation. So these can be applied to the channels of EEG, or we can use ICA, for example, to separate brain components of interest, and then use ERPs or amplitude thresholding or spectral power thresholding or mapping, basically any BCI sort of technology, either on the channels or the independent components. And then finally, we can use these coherence measures, phase coherence, phase lag index, the directed transfer function, and the partial directed coherence to measure connectivity, again, either on the channels or the independent components. And there are many resources you can use you can find on the web. In particular, EEG Lab is free software and has tutorials on how to analyze, how to conduct a lot of analysis with the EEG. Or you can also look at use the field trip software. BCI Lab is built on EEG Lab, is software for BCI problem in particular. There's a connectivity toolbox associated with EEG Lab from Tim Mullen called SIFT. And there's another toolbox for EEG and MEG analysis in Python called the MNE toolbox. And it has a lot of functions as well. And you can find a lot of EEG data and BCI tasks with labels and descriptions from this BCI Competition 4 website. And so I'd just like to acknowledge my collaborators and coworkers and my supervisor, Hirata Sensei. Thank you for your attention.
That was really excellent. Thank you so much. We have several questions. So one of the first questions is, how do you filter data in real time before sending it to a trained classification model for BCI applications?
Well, so you design a filter, basically a finite length filter, and there are tools. I usually use the Parks-McClellan filter in MATLAB, where you can design the filter response that you want, usually a low pass, a band pass, or high pass filter. And the specifications, the number of sort of decibels you want reduction in the stop bands, and it will give you a filter of a certain length that meets those specifications. So then the issue is that the filter has a certain length, which may be rather long if you have very high specifications. So then you incur a delay. So yeah, basically you're designing so yeah, some people use an infinite impulse response filter, which can be shorter, but then which can lead to instability. So yeah, you try to compromise with and get a filter as long as possible that leads to a delay that's acceptable, but has the best specifications. So if the filter ends up being too long, then you try to sort of reduce the noise reduction level, and then that reduces the the filter. And you can try different filter design methods, but that's so basic. Yeah, you're running the real-time signal through the filter, and then it comes out with a delay, and then that filtered signal goes through your subsequent processing.
Okay, thank you. We have a few more questions. What is the best way to improve the signal to noise ratio of a neural, I think neural signal in real time for brain computer interfaces?
So I mentioned the active electrodes. I think the best performing electrodes I've seen are the biosemi or possibly other types of similar active electrodes, which have this sort of onboard noise canceling technology, but they're very expensive. And so yeah, usually if you want more portable type electrodes, then you end up with more sort of noise interference, which you can then try to handle by filtering. But yeah, the best, the best approach is to have the sort of most expensive electrodes with gel, the best contact with the skin, this scalp. But if you don't have that, then like if you're not using gel, like then then the electrodes are designed to try to mitigate the noise inside the electrode itself. But I guess you need to follow the instructions, and there are different types of electrodes that want you to do different types of methods of attaching them to the scalp. But yeah, so it's, it's again also a question of what your application is. If you're going to use ICA, for any, so if your BCI application requires you to be moving, there's nothing you can really do about it. But ideally, you'd like the person not to be moving and to be as stationary as possible. But yeah, so the basic ICA model is sort of is assuming a stationarity and a sort of stability of the signals that needs to be revised or extended in the case of the person moving. I'm not sure if that answers all of the question.
So we have quite a few other questions.
Okay.
Another upvoted question was, how many IC components are standard to choose for EEG ICA analysis?
Well, the standard is you get the same number of ICA components as you have number of channels. So if you have 64 channel EEG cap, then you'll get 64 channel EEG data, and then the standard ICA model, as I said, is a square matrix, which has the same number of ICs as it does channels. So you'll get 64 ICs. I mean, then you can, I mean, that's what you'll always get. And the number of actual brain components is usually maybe 10 to 20, depending on the task and the variety of the stimuli and so forth. But yeah, so again, if you're using 128 channels, you'll get 128 independent components. But the brain components, you're not actually getting necessarily more brain components, because the brain components are limited by the I mean, there's only a limited number that are sort of strong enough sort of neural synchrony events that can be detected by the EEG. So usually the more channels you use, the more noise components. So you can sort of better fit the noise, but you won't necessarily get more brain components. And actually, you introduce more degrees of freedom, so that it can be more difficult to estimate the brain components, given all the extra degrees of freedom. So there's kind of I've found there's kind of a optimum sweet spot, so to speak, of around 64 or 64 to 128 channels is the best for capturing the sort of complexity that's present in EEG usually, and being able to separate it out from the noise. And then as I said, as you add more channels, just the sort of components get a little bit noisier, and you get more noisy sort of artifactual components. Yeah, so.
Okay. We have several more questions. I think we might have time for one more, if that's okay with you, okay, Professor Palmer. EEG has a very good temporal resolution, but why is there a constraint on the reliable spectral information we can extract from EEG, such as, for instance, most of the literature discounts anything above 60 hertz?
Well, that's because the EEG is subject to this skull. It's recording the electric field, which has to pass through the skull, unlike the magnetic field. And it so that attenuates a lot of the activity. And in addition, the high frequency activity is generally lower amplitude, so it's more localized. To see activity, it generally has to be a synchronized firing of multiple neurons, and they generally don't synchronize at high frequencies. There's a more local synchrony, which is too weak to be detected at larger distances, or through the skull at least. So yeah, and then I think there's, yeah, so it's very difficult to record gamma in EEG. But there are people who work with gamma and EEG. But there's a bit of a controversy where, you know, people are very interested in gamma as a reflection of actual, you know, brain activity, and they're interested in the coupling between gamma and theta, some lower frequency oscillations, which are very apparent in the EEG. But one of the problems is that you can get very high frequency muscle activity associated with the eyes, or, you know, other muscles, which could contaminate the EEG, and then can also be associated with, you know, behavioral activities. So, and people have, you know, claimed in literature before that there are these gamma results that turned out to be actually more just muscle activity. So that could play into the kind of hesitancy of people to accept, you know, a new result, saying I found, you know, this amazing gamma result in the EEG that, you know, they may think I mean, you need to make sure that it's not a muscle related. It's not in any way correlated to eye ocular activity or other muscles. Yeah, so you just need to be extra, you know, careful in verifying and validating a gamma result in EEG.
Okay. There are still quite a few other questions. Is it okay if the attendees who still have questions could email them to you after the workshop?
Yeah, absolutely. Sure.
Okay. I don't want to take up any more of your time. I know it's quite late where you are.
No
So
problem.
thank you so much. This was really wonderful. I'm really excited that I got to hear you speak about this. It was really
Oh, thank you very much.
insightful.
I'm
So
glad
thank you
to
so much for your
participate.
time.
Okay, so take care.